Hello friend, welcome to my channel, and today I am going to show you data representation for multicolor flow cytometry experiment. Many researchers or scientists are publishing their data in the form of SPICE diagram or pie chart with arch you can see here to show the co-expression of different markers in flow cytometry analysis. The other one you can see the pie chart or SPICE diagram showing the data representation of different markers having the co-expression on different population like CD4, T cell, NAV, Treg and Th1 cells. To prepare the diagram, you have to use the SPICE, that is simplified presentation of incredibly complex evaluation. If you visit the website, you can get the more detail about this software, and you can click here on the help, and now we will get the more menu of help where you can get the much more detail about this software, how you can use the software and how you can install this. So SPICE is only working with the Mac OS and the version 10.13 or above, and this is open source software. Here if you click, you can download the package, and this comes with the two software, PESTAL and SPICE, that prepare your data that is suitable or compatible for the SPICE. And in the PESTAL you can manage your matrix data, and SPICE is the main software to prepare the diagram. And here I'm showing the data representation or preparation in the SPICE. So you can see first is the pie chart having the arches. So maximum we can use the 6 parameter. You can manage all these things in the property panel. At the right side you can see here layout, scale, base and other things. I will show you later in this tutorial. And you can also prepare the bar figure, you can see here, and also the student t-test, CoolPlot figure, and you can also prepare the n-plot figure. So this is very important software for the multicolor flow cytometry data representation. And now I'm going to show you how you can prepare these type of the data representation of multicolor flow cytometry experiment. So let's start. So here I'm showing an experiment and the data analysis using the FlowJo software. FlowJo is very important flow cytometry data analysis software, and you can use the .fcs file generated from any cytometer. You can use that for your data analysis, and you can get the help for the data analysis if you visit the FlowJo website, learn and webinar. You can get the latest webinar about the data analysis and also the other tutorials about the data analysis. So if you visit this, you can see the link to join the list of webinars, and you can also get the recorded webinars from here. You can go through it and get the more information about the flow cytometry data analysis, acquisition and many more things you can get from here. And the other very important source I want to show you so that you can visit that and get the detailed and extensive information about the flow cytometry experiment data analysis. Here you can see the FlowJo media. This is very important YouTube platform to learn the data analysis of flow cytometry experiment. You can get each and every information about the analysis. So I highly recommend you, if you are doing the flow cytometry experiment and the data analysis using the FlowJo, you definitely visit the FlowJo media to learn the data analysis from here. Now here I am showing the my experiment, in which you can see here I have 17 sample, that is from 3 group: ASY, that is asymptomatic individual, 6 sample, endemic control 6 sample, and the VL patient, that is visceral leishmaniasis patient 5 patient. And the first thing is very important, you have to create the keyword so that you can assign the identification of your samples like this. So if you are doing your data analysis in FlowJo, you definitely know about this, but here I'm showing that how you can add this. So for this you can select the menu your workspace. In the workspace, you can see add keyword. If you click here, you can add the keyword like sample type. You will get the column and or anything that you want to assign for any particular sample. So I'm not going to do again here. So to prepare the spice diagram, I'm going to select the CD4 population from my data analysis, and I will show you the data representation only on CD4 T cell, but you can prepare any of subpopulation. For the this tutorial purpose, I am going to use only CD4 T cell from all the samples to make the spice diagram. So first thing, you have to concatenate the CD4 T cell from the samples. So better way to do this, select the CD4 from any one of sample, right click and select equivalent node, Control Shift E or Command Shift E in case of Mac. Now all CD4 are selected. Click here on the file, export and concatenate. Select the export and concatenate option. First I'm going to select the concatenate option. Output FCS3 destination, you can select your destination from here, and include event, include all, or you can restrict the number of cells that you want to use for each sample to concatenate or to make the data. So I'm going to use the 1400 from each sample to make the concatenate file, and here in the parameter you can select all uncompensated and all compensated. So I'm going to select the all compensated parameter. You can click here to expand the advanced option. I'm going to rename the file of concatenated FCS demo additional parameter. You have to select the additional parameter that you have assigned using the keyword. You can see the sample type here. You have to select the sample type from any one of your sample. So click OK and concatenate. Now I'm going to open the concatenated FCS file in new workspace from here. To close this, new workspace is open and file is present. And if you open the folder, you can see demo1.fcs file is present in the destination folder. And the next thing, you have to define the population, asymptomatic, EC, and the VL in your concatenated file. And if you open the demo FCS file and if you click here on this parameter, you can see there is an additional parameter, that is sample ID and sample type is present in our FCS data file. And now we can see there is clear cluster to define the to identify the subpopulation of asymptomatic, endemic control, and VL day 0. So I'm going to make the gate here asymptomatic, EC, and VA. Now we have to further gate to assign the number of individuals like asymptomatic 1, 2, 3, 4 like this. So I'm going to do this first for the asymptomatic. Asymptomatic 1, 2, 4, 5, 6 like this. I'm going to differentiate the individual in the EC as well. C1, 2, 3, 4, 6 for the VL. So we have assigned the individual in the asymptomatic, EC, and the VL group. I'm going to close this. I'm going to save the workspace. And the next thing you have to perform from this data, select the any individual of any group like asymptomatic, EC, or VL. And now at the Y parameter, select the histogram. And next you can select your marker that you want to make the spice diagram. And in my case, I'm going to use the CD38 first, and I'm going to make the range gate here. I'm giving the name CD38. And for the second parameter, I'm going to select the x-axis, CXCR3. So I'm showing only for the demo purpose. So I'm not going to consider the exact gating, but you can use the FMO to make the exact gate in your case. So I'm just showing the how you can perform this data analysis. CXCR5, 45, CCR6, CCR4. So you can see here I have make the gate for 6 parameter. So in Spice only you can prepare the RCS for 6 parameter only. Maybe in the future they will extend the limit of the parameters. So I'm going to show you the next step for this. So select your all the parameter, click here on the tool. You will see the option for the boolean, and you have to select the create combination gate. Click here. You can change the abbreviation here if you want to change. Like I recommend you don't use the plus or minus sign in case of any selected parameter, otherwise you will face the problem after our combination gate. So I'm going to click here OK. You can see it create the combination of all 6 parameter are selected individual on CD4 T cells. You can see here. So in this way we have to prepare for each individuals. So I'm going to select the 6 parameter. I'm going to copy the gating, and I will show you how you can apply on each individual. So select all the individual, and now you can paste the parameter gating. You can see it is applied on all the individual, and now you have to perform individually. There is no option to do in a single click, so you have to perform the combination gating for each individual. So I'm going to select for the asymptomatic 2, create combination, click OK. Like this I have to do for each individual. Please be careful. Don't leave any parameter from any individual, otherwise you will face the problem in the data matrix preparation. So combination is created for each individual. So now you have to export combination in the table. First, I'm going to export for the asymptomatic to select the combination data. Only I'm going to, so here you can see I left the for the sample one. I am going to deselect all the other things that is different from the combination gate. So now I'm going to export on table. Just select and drag to the table layout and click here on the create. Now I'm going to save the Excel file. So it's showing error, so I'm going to just Control A, Control C, open the Excel. I'm going to save this. This is a symptomatic file. I will make the tab for the next EC and UL. I'm going to make new table for EC here. Again, I'm going to select the combination data of EC individual. Now I'm going to deselect the other thing that is not in the combination way. I'm going to drag this in the table editor, Control C. In the second tab, I'm going to paste here for the EC as well. And now I'm going to create a table for VL. Drag and drop to table, create table, Control C, going to make new sheet, Control V, will be 0. So now we don't need this WSP file for the spice diagram preparation. So I'm going to close this. The main file also I'm going to close this. Now I'm going to show you how you have to prepare your exported data. So first I'm going to delete the mean and ST in the first column, and you can see here for the asymptomatic one there is the combination first, all with the positive denotation. And if you scroll down on the right side, you can see at the column BK we have the asymptomatic individual one combination, and from the BL it is no from the VM it is now changing asymptomatic 2. So we have combination up to VL for the asymptomatic individual one, and if you select all the data you can see there are 64 combination for asymptomatic one, or you can say for each individual we have the 64 combination of the parameter. So we have to break these 64 combination in different rows so that we can identify this is for the asymptomatic one or individual one, individual two. So I'm going to select the data from VM and click here on the keyboard, control and shift, and the right arrow to select all the other data. Press the Control X to cut the data, and now press down arrow key to select this cell, and now again press Control and the left arrow, and now again press Enter Control V to paste here, and now press the Control and now press the up arrow so that you can come to the first cell of the row 2. Again press Control plus right arrow. You will be here at the end of the first sample combination, and now press down arrow once and right arrow to, to come at the first combination of the third sample, and now again press Control Shift and right arrow to select all the everything. Control X, follow the same thing that I have discussed with you to make the combination of each individual like this. So you can use anything that is convenient for you. Anyhow, you have to just prepare your data in the combination form like this. So you can see here I created the for the sample accepted 1 to 6 with all 64 combination. So combination name is same for each individual. So I'm going to delete the initial and the frequency of parent part from this data. So to do this from each combination, there is a shortcut for this. Select all the everything except the combination. Control C, press Control F, paste it here, and the replace option, select all. Don't put anything here. Please replace all. You can see all done. We made 64 replacements. Every so from each column the sample naming is deleted. And now I'm going to do for the remaining part from each file as well. Control F, replace all. Now you can see remaining part of the each column is deleted. Control S. Same thing we have to do for EC and VL as well. So I'm going to do this. For the VL, I'm going to do this for the last one. So now I'm going to copy the data from the EC, Control C. I'm going to paste in the asymptomatic and for the VL as well. So other thing you have to do for data preparation is add two column here. Is in the first column use type of sample and the second the subject number. So first is asymptomatic to I'm going to provide the name asymptomatic up to 6 because we have 6 sample and the subject number like this. I'm going to put the subject number 1 to 6 for the EC, and this is very important. You will get the same nomenclature in the SPICE data that you are putting here. So please take care about this. And the remaining it is VL 5 individual. I'm going to put the number in the subject column as well. Save this, and here it is very important to save the file in the form of .txt. So I'm going to click on the save file, save. So I'm going to click here on the file, save as, browse, Spice tutorial. So I'm going to change the save as type .txt, and I'm going to rename it as a demo to save. I'm going to play this. So I was doing all these things on the Windows system, but now I'm going to move on the Mac OS in my VMware so that I can show you the Spice data preparation. Now I'm going to show you all the data preparation in the Spice in my Mac OS. Here you can see I have already installed the PESTAL and SPICE in my system and also imported the demo.txt that we have created in the FlowJo data analysis. So first you have to open the PESTAL and you can create the data for SPICE. So I'm going to select the demo.txt, click here on the open. Now you can see here left side menu and the data file here. I'm going to select the column role type is selected as category and all other things as a value, but you have to also select the subject as a category. And at the right side you can see here data rows 17, category column 2, matrix column 64, and the variable 6 that we have here. And you can see the combination as well at the lower side, matrix preview. And the next important thing, you have to click here at the matrix, and you can play with this matrix. And if you want to like remove remove any parameter here, you can remove from here according to your requirement in between the data analysis. And now I'm going to save the file, save as, on the desktop. I'm going to close this. Here you can see dot demo.spd file is generated. So this is, and this is the file that you can use for the spice. So now I'm going to open the spice, and click here on the file, open, and I'm going to select the demo.spd file, and you can click here to import the file. I'm going to provide the name demo as well for the spice file. Next, save. First query, so you can see here the first thing, you can, the entitled query, you can rename this data analysis 1. In the query section, if you click here on the plus button, there are a lot of options to create this. So first I'm going to create the pie figure for our data. So you can see pie figure is generated, but you have to make some changes here. Select the query, and here for the type, select the overlay. You can see here asymptomatic EC and VL group is separated, and the subject as a group. And now click here on the pie figure, and now you can play with the setting the pie figure. Like if you want to make the layout and appearance changes, you can make here like radius. If you want to increase the radius, you can increase, and the margin also, and scale and base and arch notation. If you select the arch, you can see the arch of parameters 2, 3, 4, 5, and 6. And legend, if you click here on the legend, you can see the arch legend as well, color representing the particular marker. And if you click here on the category, you can see the category as well. I'm going to deselect this, click here on the overlay, asymptomatic EC and VL, and group. Whatever you want, you can select here at the legend that will show in this pie figure. And further, if you want to make the changes like for the arch legend color or arch color, you can select here colors and you can make changes from here. Standard, and you can manually add any color if you want to change. Select any color from here, and like if you want to make it black, just select the black. The first color for the CCR will be black. So in this way you can change the color scheme as well. There are predefined color scheme like standard and other. So I'm going to close this. And if you want to make changes in the arch, so you can see the thickness and spacing as well. If you want to make more thick the arch, you can select this thickness option, and the spacing, if you want to make the space, you can make the space by the spacing option. For the pie chart, you can also make the color changes or apply the scheme of color like gray to black. You can select here. So in this way you can create the pie chart along with the arches showing the co-expression of different markers. You can see for the asymptomatic EC and VL. And next thing, you can also add the bar graph or figure. And you can see bar graph is created, and you can make changes in this bar graph. You can see the option available for the bar graph. I'm not going to it in a greater detail, but you can see there are a lot of options. You can make changes in the bar graph. So you can see there are a lot of options available. Error bar, whiskers, point bar. So you can go through it and make necessary changes required for you. And next thing I'm going to show you. And next thing I'm going to show you add gen plot figure. You can also add this figure. You can see here, and you can also make some changes in this figure. And the other thing I'm going to show you the cool plot figure, permutation test, student t test. You can change the overlay category from here. In this way, we can do many things using the SPICE to represent our flow cytometry data. So I'm not going to greater detail in this, but I just wanted to show you that you I just wanted to show you that how you can prepare this and present in your publication. And if you want to know more, You can comment in the comment section. I will try to explain each and every graph in the detail so that you can understand easily. But anyhow, if you want to more anyhow, if you want to know more, you can also visit the Spice help present at the website to more about this, to know more about this. And the last thing I'm going to show you that how you can export this data file. So I'm going to select the all the thing here. I'm going to export. You can choose the destination folder. Here you can see. Here you can see demo folder is created and all the plot is present in the form of PDF, bar graph, 5 figure. So these are the vector format plot. Like you can use third party software like Illustrator to further edit the content to make better for publication purpose. So in this way you can create the spice plot or pie chart data from the multicolor flow cytometry experiment. So I hope this may help you in your research and publication. And thank you for watching my tutorial. And if you like the video, please subscribe my channel. Thank you.